Now, there are a whole host of factors that can contribute to herb-drug interactions, and of course, the more botanicals or the more phytochemicals you're exposed to, the greater the likelihood that there may be an interaction. And of course, a lot of products that are on the market now contain more than just one botanical extract. Oftentimes, it's a multitude or multiple extracts in one particular product. Of course, you have to realize that these are botanical extracts. You're taking the botanical material, extracting it with either hot aqueous or hot organic solvents, and concentrating a lot of these phytochemicals into a form that's much higher than it would if you just, say, consumed the plant on its own. And of course, a lot of the phytochemicals that are in some of these interesting and unique and sometimes quite varied products, we really don't know much about their pharmacological activity. All right? And so an example that I like to use is that there have been over 500 unique phytochemicals identified in Panax ginseng. And just about every multi-botanical supplement has Panax ginseng in it. And so if you think there's 500 or so in Panax and you've got 6 or 7 others, then it's a multitude of compounds that are in those things. And so this is another slide showing just a partial listing of some of the known constituents in St. John's wort extracts. And so several phytochemical families, so to speak, are fairly common in most botanicals, like the flavonoids and essential oils. But every now and then you run across some unique phytochemicals that are found only in one particular species. And a good example with St. John's wort are the naphthodianthrones, over here on the far left, and then the floroglucinols, and one in particular, hyperforin. And we're going to talk about hyperforin in a little more detail later. But you can certainly see that when you consume a St. John's wort product, you're consuming just more than one single entity as you would with a conventional medication. Now, in terms of recognizing or our initial recognition for the possibility of an herb-drug interaction, most of the time we're initially alerted to a potential herb-drug interaction through case reports that appear in the medical literature. Now, of course, case reports really can't establish causation, but they can certainly help generate hypotheses about possible mechanisms. But if you start to see the same types of case reports over and over and over reporting the same thing, there's probably some merit to that. And that was certainly the case with the ephedra products that we'll talk about in just a bit. And of course, this also holds true for the adverse event reports that get submitted to the FDA's MedWatch program. And of course, if you're into this area like myself, the literature is replete with in vitro studies assessing whether or not phytochemicals, if you expose either purified drug metabolizing enzymes or transporters or cell lines that express certain drug metabolizing enzymes or transporters, if you expose them to a high enough concentration of a purified phytochemical or to an extract, you're going to elicit some type of response. And oftentimes, to do that, you have to solubilize some of these phytochemicals with solvents like DMSO and such. And so a lot of the in vitro studies almost always imply that there's going to be some type of adverse effect. But rarely do those in vitro situations translate to the in vivo condition. And so to me, just my personal opinion, is that the most definitive way to assess whether or not a botanical is going to lead to an herb drug or clinically relevant herb drug interaction is to do prospective clinical studies in human volunteers or in actual patients themselves. And so we're going to look at some examples of all of these as we go along.